Salk Institute researchers, as part of a larger collaboration with research teams around the world, analyzed more than 500,000 brain cells from 3 human brains to assemble an atlas of hundreds of cell types that make up a human brain in unprecedented detail. The research, published in the journal Science, is the first time that techniques to identify brain cell subtypes originally developed and applied in mice have been applied to human brains. The researchers say that this is really the beginning of a new era in brain science, where they will be able to better understand how brains develop, age, and are affected by disease. The new work is part of the National Institute of Health's Brain Research through Advancing Innovative Neurotechnologies Initiative, or the BRAIN Initiative, an effort launched in the year 2014 to describe the full plethora of cells as characterized by many different techniques in mammalian brains. Every cell in a human brain contains the same sequence of DNA, but in different cell types, different genes are copied onto strands of RNA for use as protein blueprints. This ultimate variation in which proteins are found in which cells and at what levels allows the vast diversity in types of brain cells and the complexity of the brain. Knowing which cells rely on which DNA sequences to function is critical not only to understanding how the brain works, but also how mutations in DNA can cause brain disorders, and relatedly, how to treat those disorders. In the year 2021, the Salk team profiled 161 types of cells in the mouse brain based on methyl chemical markers along DNA that specify when genes are turned on or off. This kind of DNA regulation, called methylation, is one level of cellular identity. In the new paper, the researchers used the same tools to determine the methylation patterns of DNA in more than 500,000 brain cells from 46 regions in the brains of 3 healthy adult male organ donors. While mouse brains are largely the same from animal to animal and contain about 80 million neurons, human brains vary much more and contain about 80 billion neurons. Since the new Salk research was intended as a pilot study to test the efficacy of the techniques in human brains, the researchers say they can't yet draw conclusions about how many cell types they might uncover in the human brain or how those types differ between mice and humans.